Hi, my name is Julissa Rodriguez, and this is my protein project part 2. The protein I will be showcasing and talking about today is MC1R, known as the melanocortin 1 receptor protein. It is a type of G protein found in the plasma membrane of melanocytes, which are pigment-producing cells in the body. Normally, MC1R produces eumelanin, which is dark pigmentation responsible for darker hair and skin, but when the R67 mutation, commonly seen in beach mice, occurs, it results in lighter coloration of the skin or hair. And this happens because of amino acid number 67, arginine, which is seen here, is replaced by cysteine, which I produced a different model showing the mutated version. And here you can see how arginine is replaced with cysteine. I used popsicle sticks to represent the cell membrane, and I used pipe cleaners to mimic the spiral shape of the MC1R protein. And as you can see here, I added the intracellular and extracellular loops that MC1R contains. I also used clay to show the 7 transmembrane alpha helices that you can see here in red. And then I used circular clay balls to represent the amino acids forming this protein, which are right here. And then I added a 3D representation of AMSH using construction paper. My protein does not contain any subunits, but it does have a primary, secondary, and tertiary structure. I chose to display the primary structure by using the circular clay balls in orange to demonstrate the amino acids that make up this protein. The second structure is shown through the rectangular red clay right here that demonstrate the transmembrane helices, and then the overall 3-dimensional shape of my model and curvature / folds and interaction showcase the tertiary structure. Arginine R is a hydrophilic amino acid. Right here you can see arginine, and it's positively charged because of its guanidinium group, which forms strong interactions with water and polar molecules. This property allows arginine to play a crucial role in protein stability and function, especially through hydrogen bonds and ionic interactions. When arginine at position 67 in the MC1R protein is replaced by cysteine, a hydrophobic and polar amino acid, and sulfur-containing side chain, it disrupts the protein's normal functions. Cysteine lacks the positive charge that arginine contains, so it can't form the same hydrogen bonds or ionic interactions that stabilize the receptor structure. This could lead to altered protein folding and reduced function of the MC1R receptor, potentially resulting in changes like altered pigmentation, which occurs in beach mice. This occurs because normally AMSH, which is here in our non-mutated version, binds to MC1R, triggering the production of CAMP inside the cell. CAMP then activates a signaling pathway that regulates melanin production. However, the cysteine substitution that you can see here leads to a reduced CAMP production and impaired signaling, resulting in changes in skin or hair color, as we see in beach mice. Here is a clear view of the mutated version. And then This is the regular protein with no mutation. 3 limitations that my project includes is that, first off, it shows static structures. It cannot fully replicate the dynamic interactions of the MC1R receptor with ligands like AMSH or the downstream signaling pathway like CAMP, which I wasn't able to show. And it doesn't show how MC1R changes shape upon binding to AMSH. Another limitation includes the size of my project, which is much bigger than the actual MC1R, which is extremely small. Lastly, it doesn't accurately depict the interactions that occur in secondary structures such as the beta sheets and alpha helices interacting. Well, that was the end of my video. I hope you guys liked watching.